This video is on enzymes, which is quite a big topic in biology, and we'll go through it step by step, but really there's not a lot in terms of the student learning outcomes. So the main thing here is that enzymes are specific for their substrate, increase reaction rates by lowering activation energy. So there's a few different terms there you need to understand to be able to go through, as well as being able to describe the induced fit model of enzyme substrate binding. So enzymes are proteins and they have special roles in the cell, the main one being here acting as a catalyst, which is also related to their ability to increase or control the speed of reactions. So a catalyst is a substance that participates in a chemical reaction but isn't created, consumed or changed during that reaction. So what we have in a chemical reaction is our substrates that are turned into products. The catalyst helps with that, but it's not a substrate or a product of that reaction. So the key thing is just understanding how this might work. So here's an example of a substrate becoming some products there. So this shape is broken into two things representing one molecule where the bonds are broken to become two separate molecules. As you can see, the enzyme is involved in doing that, and at this intermediate step forms what's called an enzyme substrate complex. The substrate binds to the enzyme briefly before that comes apart, and then the substrate is broken in two. So it's important to understand how this works, but the substrate becomes the product and the enzyme helps that. So we're going to step through some of those details. Before we get further though, you need to have a general understanding about the way chemical equations are written. The substrates are always on the left, and we put a plus sign between each of those. The products are on the right, we put a plus sign between each of those, and there's an arrow in the direction that the reaction is going. So the general sort of form is something like A plus B becomes C plus D. We might only have one substrate that gets broken down. This is called a catabolic reaction. So A becomes B plus C. Or we might have an anabolic, a synthesis reaction, where two things are put together, A plus B becomes C. And they're the general kind of ones. So here's an example of a catabolic or a breakdown reaction. And in fact, this is the one that we're going to use for our practical. It's the breakdown of hydrogen peroxide into water and oxygen by the catalase enzyme. So when we look at this reaction, we can see a number of things here. Substrates on the left, products on the right. The enzyme is written over that arrow. These products are separated by the plus sign. Each of these is written with their chemical formula. H2O2 is the formula for hydrogen peroxide. Chemistry 101, that means it's got two hydrogen atoms and two oxygen atoms. This two out the front means that then there's two molecules of hydrogen peroxide, becomes two molecules of water and one molecule of oxygen. So that's one example of one reaction, but actually each enzyme is specific and catalyzes only its specific reaction. So this enzyme number one might use these two substrates in an anabolic reaction to make something new. Enzyme 2 uses a completely different substrate, and these substrates don't fit with this one. Enzyme 3 is different again. So each enzyme is specific for its substrate and is involved in its specific reaction. One of the reasons why that is we start to see with these shapes here, is that each enzyme has what we call an active site. The active site shown here is based on the folding. So here's our polypeptide folded shape into the tertiary structure of this enzyme. If we use space filling instead, we can see all the atoms that make up that enzyme, and there's a gap in here that's called the active site. The substrate fits in there. We sometimes think about a key and lock model. But actually it's more than that, because the protein itself can fold and change, and depending on the binding of that substrate will actually perhaps close around that or bend in some different way, and that's what we call the induced fit. So the fit is made even more tight by, and that's induced by, the binding of the substrate. That's really important to its function. So the next key concept we need to look at is activation energy. So just to define that, the enzyme substrate binding can weaken bonds or force them together. So if we're breaking this substrate up into two parts, or putting two parts together to make a new product, that requires making and breaking of bonds. And that's where this enzyme substrate binding is really important. Because what would happen is that without an enzyme, this substrate would have to break up into the two products. And although that can happen, that sometimes the substrates become the products without the enzyme there, that's a very slow process, and it can only happen when there's a lot of activation energy. What the enzyme does is allows that forward reaction to happen with much less activation energy, because this binding can help, in this example, to weaken those bonds to break that up. And so one of the key things that enzymes do is that they lower the activation energy. And that's why those reactions are faster. This is the way we represent that on a graph. So without an enzyme, and this reaction here is the breakdown of glucose and oxygen into carbon dioxide and water, you should recognize this as the respiration reaction. So the reactants have this amount of energy, and the products have this amount. So you can see that there's some energy released during respiration, and that's important because that's why respiration is so important, is it releases energy. And so it's an energetically favorable reaction. So that should be fairly easy to go down this way. But before we can roll down the hill and gather that energy that's required, we actually have to kind of go uphill for a bit. So in order to break those bonds and move up here and then reap the reward from the energy released, there's actually this hump to overcome that's called the activation energy, and that's quite high. So if you have glucose and oxygen, a bowl of glucose sitting in the air with oxygen there, it will react very, very slowly, and not much of it will break down to being carbon dioxide and water. So the glucose can sit there for a very long time before we ever get that reaction going forward. Whereas in the presence of enzymes inside your cells, the oxygen and glucose can react much more readily. So that with the enzyme, you can see that there's a big loss of activation energy, which means, or reduction in the activation energy required, which means that this forward reaction can happen much more quickly. So how does that happen? Well, here's another example of lactose being broken into glucose and galactose. Again, this is two sugars that break up to form those, and this man pushing the ball up the hill is meant to represent the high amount of activation energy required before that ball can roll down. In this case, with the enzyme, what we see is that by binding and forming an enzyme substrate complex, that the enzyme changes shape around there and actually puts strain on those bonds and will break that bond and mean that the glucose and galactose can come off more easily. So by binding to an enzyme, that you are putting strain on the bonds, and that lowers the activation energy. So this is what we've got here. They can orientate substrates. So for instance, if you have two things you want to put together, if they're being held in place by the enzyme, they're much more likely to line up those bonds, those two atoms that are bonding together, rather than just random collisions. So they can orient substrates and therefore make products happen much faster. They can put a strain on bonds to break them, or they can stabilize intermediate states. So enzymes help to lower activation energy by one of these three means. This is covered in Chapter 3. There's some videos there. A video on the catalase experiment that we're going to be doing next week. Because catalysts and enzyme rates is a major topic in chemistry, some of these videos take the chemistry angle rather than the biology angle, and so if you're doing chemistry as well, they'll be useful, but if your chemistry knowledge is a little bit shaky, they might be a bit full on for you. But give them a go anyway.